Thank you for the introduction. Good morning, everyone. Thank you for coming this early. I'm from City University of New York. This is a joint project. It's a very long project, has been going on for years, and it's still going on. So we have a very long list of students and collaborators who have worked on this, including Pengxiang Wu, Mingchen Gao, Shaoting Zhang, Zhen Chen, Changhe Yuan, Yusu Wang, Dimitri Metaxas, and Leon Axel. A little bit words about me. In my previous life, I worked on algorithm design and complexity analysis for computational topology problems, as well as algorithm to compute persistence more efficiently. Now I switch to the evil side, start working more on the application side. Okay, so today I would hope to introduce one particular application where, to me, it's a very convincing example of how persistent homology, in particular something more than just zero-dimensional persistent homology, that can help us in practice. So our particular problem is cardiac image segmentation. So the input is a 3D scalar function, which is the image of the human heart. And the goal is it can come from CT image and MRI image and sometimes even ultrasound. In particular, we're more focused on the CT image, which has higher resolution. So you can see a lot of details inside it. And this is actually the left ventricle of the heart, and this is the left atrium. Okay? So the goal is, with this input function, can you find a 2D surface, separate the space and blood inside the ventricle and outside. Basically, you want the inner surface mesh of the ventricle. That's a goal. Okay. So, but the problem is very challenging. And actually, here's what's really look like inside the ventricle. You can see it's actually like a sponge with a lot of very fine muscle columns. These muscle columns are called trabeculae. And here's a real view, a view of actually a live heart. They just came out of the human body and they're still alive. So you use a lot of the electrical stimulation to make them still beating. And unfortunately, the video is not working. But you can actually find those kind of interesting videos from this online website where you can see a lot of videos. Okay, and it's very important actually to figure out exactly how these things look like, even though they look very tiny, right? So what actually happens is, in practice, when the doctors try to look at a patient with potentially cardiac disease, it's very important to measure the volume of the ventricles when the heart is completely relaxed and when the heart is contracted. So this is the ratio of the volume is called ejection fraction. So basically, how much blood you are pumping out from your ventricle when the heart contracts. And if the ratio is low enough, then you have a trouble, okay? Because the blood is not flowing, not well circulated, right? And also, there are a lot of several diseases that particularly are relevant to the morphology of this trabeculation. One disease called non-compaction. So it's like genetic disease where the even in adult body, this kind of spongy trabeculation is overexpressed, and this is actually it's kind of a rare disease, but it will be very dangerous. As well as you can predict, estimating the right shape and the motion of the heart would help you predict whether this person has a risk of getting stroke and so forth. And also, desynchrony is whether the different cardiac chambers are beating in a synchronized fashion. And also, you want to actually simulate the blood flow inside the heart to actually get a better understanding of the physiology of it. All of them depend on a very accurate estimation of the surface of the ventricle. So how are we going to do it? Actually, it's very challenging, first of all, because all these fine, so this is a one slice of the image, and this is what we actually try to enhance so you can see something. In particular, you can see these are these dark bands are exactly the trabeculations we're trying to extract, right? But they're very difficult in the sense that the intensity are not uniform, and there are a lot of noise. And their shape, in practice, you can see, actually from the real image, you already see the shape and the size and the thickness, all kinds of things are different. So how can we deal with this in a robust manner, right? First thing we tried was traditional segmentation, image segmentation methods. It's been done so many years in computer vision, image analysis. So how about it, right? So what we do is we use a standard image segmentation technique. And this is a very standard setting, namely for all the pixels in the image. This is a 2D image, then it's pixels. For 3D, it's voxels. So for those pixels or voxels, each of them, we want to give them a label, either 0 or 1. Okay? If it's 1, meaning this is the object we're interested. If it's 0, meaning it's a background. So for this case, hopefully, we will find such a labeling so that we get the object singled out correctly, right? And usually, people set this up as an energy minimization problem. So you define certain kind of energy term. And this energy can spit out a scalar value for every possible labeling, every possible 0, 1 assignment to all the pixels or voxels of the image. And then you have an efficient algorithm to find the one with the smallest energy, and you're done, right? There are very many well-established models for it. But a common theme of it is it comes with 2 terms. The first term is a data term. Okay. Second term is the model term. The data term basically want to make sure the way you assign the labels at every local location is somehow faithful to the belief you have. For example, in this case, what we are trying to do is we want to set the foreground to be the muscle, so it should be the darker region, and then background to be the blood inside it should be the brighter region, and then you can assume that the muscle intensity follows certain kind of single one-dimensional Gaussian distribution, and then the blood follow another one-dimensional Gaussian distribution. Then, if you really just want to at every pixel optimize it, you find a global threshold, right? And then everything below it is exactly the foreground. Everything above it is exactly background. This will give you an optimized way to decide the whole thing, right? Unfortunately, that's not sufficient because you will see some regions you get it right, but some other regions you get wrong, and the boundary is very noisy, right? So that's why we introduced the model term. Model term usually regularize the result, try to make the segmentation result to be smooth in general. And then hopefully you can get something more reasonable, right? But you can clearly see right away that when you make it reasonable, when the boundary looks smooth, you actually completely get rid of any hint of there was a trabeculation. This is what we care about in this case, right? So the reason being, this model term takes some kind of integral over the whole domain, and they don't know when to ignore the fine details or when to keep the fine details. And all those fine structures are dealt with, like any other noise, they're removed. Okay, so what we do is we take a topological perspective, and of course we do persistent homology. So following the ritual, I'll spend 2 slides on it, reviewing. But I have a different focus in the sense that, to me, most of the function I deal with is either image function or density function. Whereas when you talk about Rips complex, check complex is more about the distance function in the high dimensional space, right? So I'm using I'm actually doing this upside down. So basically, I'm using the density function and then taking the super level set, right? And actually, this is a mistake in the sense that later on, I'm actually using a sub-level set. So please forgive me for the mistake. And so what we do is we take a point sets in 2D, and then we estimate some kind of density function, right? So this density function will give us, you can basically see the topology. You have 2 components and 1 closed loop, hopefully. But of course, the image is noisy. It's hard to figure it out. And so we use the persistence, of course. What we do is we take the super level set, we put this image into 3D so that the height is exactly the intensity. And then we cut it at different thresholds. If I cut at one particular threshold, what's remaining is a super level set, right? So the whole idea of persistence is if you take each super level set, one slice, you don't always get the right structures. On some of them, you get the right structure on some parts, but not all of them. The whole point is take the whole stack of it, right? And then, in general, you try to find the structures that are most common among these stacks, right? And this measurement of the how long it exists is exactly the persistence. So you can do some kind of filtration. You move the threshold from top to down. During the process, you can see a lot of topologic structures appears and disappears. And then you record them down. That gives you exactly the barcodes, right? And then you can transform the birth time and death time into a 2D coordinate system. Then you get the persistence diagram. In this case, I will have a lot of blue dots corresponding to zero-dimensional homology, and one red dot, purple dot. That give you the one-dimensional homology. Right? They corresponding to a long bar of 1D and two long bars of 0D. And then they gave me a hint of, okay, I have two connect components and one closed loop, hopefully. All right. Of course, it's never. Most of the assumptions doesn't quite work in practice. You have to deal with it in different ways. Okay. So this gives us a robust way to actually figure out this underlying structure of very noisy observation, right? And that's our hope. In particular, in our cardiac data, what we do is we take the image as observation, and we try to figure out the one-dimensional structures hidden in it, based on persistent homology. Then we explicit force them to be in part of our segmentation, before we apply any segmentation methods. That's the basic idea. So here is the persistence diagram of the 1D of this 3D scalar function, and I'm just singling out one particular sublevel set in this case. As you can see, in this particular sublevel set, some of the handles are already appearing, right? And of course, there are other handles that go through the slice, so you can directly see it. All right, and this. As you can see, this persistence diagram itself is still very noisy, very bad. A lot of dots, and most of them are really not the signal we are looking for. So we empirically select a particular threshold of persistence b equal to 80, just to rule out a lot of negative ones. By the way, the red dots are negative ones, blue dots are the positive ones, the ones we want to keep. And this is based on human observation. That's why we have students actually look at the images and try to label each of them. Are they a true signal or bad, or really the artifacts from the noise? Right? So only using persistence threshold is still not sufficient. You can see within this threshold, we still have the red dots and blue dots. So what we do is we further plug in these handles into a machine learning classifier, basically a simple classifier based on geometric features. And then use these geometric features, the selected geometric features eventually are the handles we want to keep. Okay, that's one of the messages I want to, in general, I always try to bring up, in the sense that to me, topology is not the golden rule to select the right thing. Persistence is not necessarily the golden rule. The fact is persistence and topology help us to find the singling out a finite number of good hypotheses that we can work with. And then we can start from those handles and topological structures, try to extract the interesting features, and use those to maybe fit it into some classifier or other cases to start it. To me, that's, in practice, that's usually where it works. Okay, so once we selected these handles, in particular in this case, I can see these two blue handles. Since I'm taking a 2D slice, so part of the second blue handle was occluded, right? And And then we force them to be part of the final segmentation. So that's why we would have the result that has the trabeculation. Okay, just to compare, this is the original result using a standard segmentation method. We explicitly enforce these handles to be part of it. Okay, so here's a zoom-in observation comparing the two results. As you can see, this is a clear example where the mesh, the handle is missing, and our method is able to reconstruct it. Okay, so that's nice. Are we done? Of course not, right? So, one more problem. When we finish this, we notice certain things that are weird. Okay, what are the strange things? In particular, some of the handles, of course, in this case, we're successfully reconstructing the handles in the middle, but what we reconstruct is really a weird shape. It's like a spider, but that's not what it's supposed to be, right? Because when you look at the real image, okay, the handles are mostly stretched. So it's like a sponge or mesh that you maximally stretched. Of course, this is only when the heart is fully relaxed in this case. And then I will talk about it later when we start working, at which stage of the heart we work on. Okay, so this is what really is supposed to be. Okay, so how do we handle this problem? And this is not a very rare case, so we really need to deal with this. So we looked into the problem and realized the following. In the persistence context, you detect the existence of certain handles, but you are never told what exactly this handle is. There's a way to do it, of course. In particular, you can extract certain columns of the reduced matrix that give you the cycle. But that's not necessarily the unique one. In particular, for these handles, I can have the yellow cycle, I can have the green cycle. All of them can represent the same handle. And of course, you know which one you like, the yellow one, right? But I intentionally drew this one really bad. So, I mean, there are cases where it's arguably this one or that one, right? Really depends. So the basic point is it's not unique. You need a way to select the right ones. And in particular, we formulate the following problem. Among all the cycles that can represent this handle, a particular persistent dot, or a bar in the barcode, we want to select the one that is shortest. Okay, at least in this scenario, this is some scenario we prefer. In general, because the whole motivation was to find the cycle representing the fully stretched. So the shortest one would, in general, try to be in the maximally straight and stretched, right? And of course, we need to formally define what they are. So the cycles we select from are the set of cycles that are created at the birth time of this dot, and those cycles that become trivial at the death time. To give you an example, okay? So this is a original complex. When it enters, this is a filtration, sorry. When it enters time B, the birth time B, we have a blue edge. Okay, this blue edge will create a bunch of cycles. This one, this one, this one, and in fact, I can create this one plus this one, right? They're all cycles that are created with the entrance, the grand entrance of the birth age. Okay, as time goes, some of the cycles actually becomes homologous to each other, but they're not what we those events are not irrelevant. What we really care is in the death time, when this so let's say this is a simplex. When it comes in, this homology is killed. This is the death time. Which of those cycles that are created at the birth time that becomes trivial are the smallest one? In this case, this is a cycle that is legible, and this is a cycle that is a good candidate. But this cycle, which was created, is not part of it, right? So we only care about these two, and maybe I need something between them. And then we will definitely select this one as the shortest one. So that's what we want to do, the goal of it. Okay. Actually, there's a way to do it very conveniently if you are familiar enough with persistent homology algorithm. And luckily, I have a bunch of lectures before me, so I assume at least you know when you do persistent homology, in the end, you would end up with a fully reduced matrix called R. That's all we need. Okay, so in the fully reduced matrix R, I mean, this matrix is not unique, but these columns, each column has the lowest non-zero entry, and then this corresponds to the birth time of a persistent dot, and then the column index itself gives you the death time, right? So basically, each of these columns give you one dot, and their lowest entries are unique. Now, if the green one is what we care about, is the class we try to localize, what happens is you can have a lot of information out of it. Okay. First of all, we take all the columns, including the one before it and the ones after it, and the ones, but only keep the ones that are shorter than it, right? So their lowest entry is above the birth time B. And these columns will give us a cycle basis, a basis of all the cycles at the complex at time B. And there's an assumption. We assume the image domain itself has trivial topology. It doesn't have any essential homology. Otherwise, we would have to attach a few extra columns after it. Okay. So next, we look at the columns, those shorter columns before the green one. And actually, these columns are the set of create the set of all the cycles that is in the complex of B, and also that becomes boundary before the time D already. Okay, but at time D, this column becomes boundary, and all the purple ones are the ones that will remain unbounded at time D. Right. So what we really care in this case is that we can show that the set of cycles that we care about that represent this homology class is really the green column plus any linear combination of these orange ones. This really orange is brown. Okay. So furthermore, we have to actually reduce the problem into a problem called homology localization that has been well studied. And I myself has been studying it in my previous life. Okay, so what we do is, if you can establish a certain simplicial complex, okay, and you fix a particular homology class, 1D, and then you want to find the shortest representative cycle of that particular class. How do you do it? Right? So there are a lot of algorithms, and quite many settings are very hard to compute. But there are settings that are doable. Okay. In particular, we are reducing into this setting where the 1D class, where the class is one dimension, and also the Betti number is bounded. Then we have a reasonable algorithm. Okay. And in this case, we can actually reduce our current problem into that setting. Remember, what we care is among all the cycles that can be represented by the green one plus a linear combination of the purple ones, okay, we want to find the shortest, right? So we can construct a simplicial complex, basically using the one skeletons of this boundary, this complex of birth time B, and that, and basically all the colored columns are the cycles of that complex, and then we make some of them boundaries, in particular the orange ones, the boundaries, and then the green and purple ones form a homology basis of that complex. And then all we care is the particular class that's represented by the green one, and then we just localize it. And then everything is solved, right? Exactly the 1D homology localization problem.
What do you mean by localizing?
Localizing meaning finding among all the cycles belonging to the same class, the shortest one. Right? So somehow the shrinking effect meaning localize exactly where it is. And there's a rationale behind it. Remember, we had the assumption that the Betti number has to be bounded to make it work, right? In this case, it's actually not a bad idea, bad assumption. Why? The green ones, remember, the green ones that we really care are the ones with reasonably high persistence. And furthermore, if you look at each purple column, this purple column is dies after this D time, and it's born before the B time. Okay, what does that mean? If I look at the persistence diagram, and if this is my green dot, right, and all the purple ones will fall exactly into this upper left quadrant. They all have a bigger persistence than the green one, right? I can assume reasonably that my threshold is good enough that the ones above a certain threshold are reasonably finite. And later on, I will show you that's actually still too big of assumption in practice. Okay, with that, now we can use the algorithm, right? So the algorithm is well established, well studied, basically. I'm using one particular algorithm, excuse me, that myself, Yusu, and Tamao, and Sergio, and a student, Busai. I forgot the name, sorry. We developed this particular algorithm to solve this 1D homology localization problem, right? The idea follows the annotation. First of all, we try to annotate the edges of a simplicial complex. Each edge will be given an annotation which is length G binary vector, and that signals how this edge is relevant to the homology basis. Okay, this is not unique, but you have to give an assignment to all of the edges. And the requirement is, if you follow any cycle in this simplicial complex, and then sum up all the annotations, the result annotation will help you identify the homology class of this cycle. Right? For example, in this case, if I follow this cycle, and the ones without annotation are just zeros, then I will have a cycle with annotation 1, 0. If I follow this one, I will get 0, 1. And if I follow this cycle, I would have 1, 1, right? So that they all give me the right homology class. This is the annotation that we can compute. And the computation time is the matrix multiplication time, where n is actually a complex size. Okay. Once we have this annotation of the complex, what we can do is we build a covering graph. The covering graph basically make 2 to the power of g many copies of the original graph. Okay, in this case, I have 2 homology, my Betti number is 2, I will have 2 to the 2, which is 4 copies. And the edges are not lying within the same copy. Some of the edges will go cross copies. And the way they go cross copies is based on their annotation. Okay, once you construct this covering graph, okay, whose size is actually n times 2 to the power of g. Now, the problem of shortest cycle is exactly reduced into a problem of shortest path problem in this covering graph. In particular, I take any vertex to start with, and I find the shortest path to the same vertex in another copy. In this case, 1, 0. I will find the shortest cycle within the homology class of 1, 0, starting with 1. Sorry. And in the original algorithm, I have to start with every vertex and then do it. But in our context, we are somehow blessed in the sense that we know our cycle has to go through the birth edge. So we actually have a way to find exactly the starting point and the destination time, the top point. So it becomes a single source, single destination, shortest path problem on this super huge graph. That's our problem. Right? So then we have the complexity, which is the graph size times the log of the graph size, which is n to the 2 to the power of g times log of n to the 2 to the power of g. So we're solved, right? Since g can be assumed to be a constant, of course, we're done. Right? Not yet. So of course. The problem is, in practice, what happens is n can be millions. Our image easily can be hundreds to the cubic many voxels, right? So you have millions of pixels, meaning millions of vertices in the complex. That's the first thing. Second thing is, even the Betti number can be up to hundreds, right? Remember that diagram, right? No matter how we select the things above it, still is a big mess. So the numbers are really huge. I mean, think about 2 to the power of 100. That's really a big number, right? To mathematicians, maybe not. Now, so in practice, it doesn't work. Actually, this algorithm homology localization have been developed in a theoretical community for many years, but no one actually implemented any algorithm along that line, as far as I know, right? We actually implemented it, and it was horrible on this complex context. On the simple one, yes, I can do a simple complex with 2 holes. That's nice. It works, right? But on our application, it doesn't. It never finishes. How do we do it? We realize we can use the A-star search algorithm, which is actually a very classic method when you try to accelerate the shortest path problem. Right? The idea is, when you try to find the shortest path, sometimes you don't have to exhaustive search the whole graph. Ideally, you only need a small portion of it before you find the final destination and get figured out the shortest path. Right? And so here is the basic points of this A-star search. You need a certain heuristic function. This f of any vertex v in the current graph is the sum of g of v and h of v. G of v is the actual shortest path from this particular vertex to the source, to the starting point, which you already know. And then h of v is an optimistic estimation of how far are you from the destination. Okay, you have to guarantee that it is the lower bound of the actual path, shortest path you can do. Once this is established, at each time you just explore the new vertex with the smallest fv, function f. And then you are guaranteed to find the global optimum when you reach the destination. All right, hopefully you'll only explore a small portion of this graph. And there's no complexity guarantee. So in the worst case scenario, you're going to spend the exact same time as the original method. Okay, so the only thing that remains is how do we design this particular function, heuristic function. Okay, we need to use the insight of the topology again. So we designed the following heuristic function h. It's a maximum of g many other small h, right? Hi. The hi actually is the shortest path cost estimation. The actual shortest path from the current vertex v to the destination, with the guarantee of the correct homology only on one particular bit. Remember, the whole thing is an annotation of g-bit vector. So when we actually try to get from the source to the destination a particular copy, we are trying to guarantee the thing to be correct on all bits of the annotation when we sum up the edge annotations. But in one particular heuristic function, we say, okay, I don't care about other bits. I only say this particular bit you have to be correct. And depends on, so I'm gonna end up with a smaller graph that only has 2 copies. Okay, And if you look at this, it's really the projection of the original graph onto one direction. Basically, I collapse this one to this one, collapse this copy to this copy, and the cross edge from left to right, and from upper left to lower right, and from lower left to upper right remains the cross copy edges. Another edge will be collapsed into the same copy. This is one way of doing it. Of course, the other way is I do horizontally, right? This gives me two of those two copy graphs. Each of them actually give me a guaranteed estimation of the shortest path, not estimation, actually, the actual shortest path cost from any vertex to the destination with a guaranteed bit number that I care about. Okay, and of course, computing shortest path in this subgraph is cheaper, right? It's actually n log n, because the whole graph is just 2 times n size, O of n log n, of course. Then the whole computation of the heuristic function overall is only linear to the Betti number. There are other things that we have to implement, some other heuristics we have to implement in practice, including we can't just get all the annotations from one shot. Remember, the annotation computation is n to the matrix multiplication time, n to the power of 2 point something. Okay, that's already huge when n is a million. So what we do is we do a lazy evaluation. We only evaluate the annotation when we have to. That's one thing. Hopefully, we only need to explore a very small neighborhood of it, right? Furthermore, in this case, we need to enforce certain kind of randomness. So the annotation generation depends on a spanning tree, which we can generate randomly. And also, it depends on the homology basis from the reduction result of persistence that we can also mess up a little bit using randomness. Okay. With all those tricks, we are able to achieve a reasonable performance in practice. All right. Here is a scalability experiment of a very simple synthetic example. This is the data, and then we try to localize this particular homology class, okay, of the persistent function of this. And then we make the function more and more complex, namely the purple columns, when we try to localize this class, actually keep increasing. That's a basic idea. The image is always the same size, basically 400 squared pixels. And then we can see the time complexity, the time we spend with the Betti number getting bigger. And the red one is the original exhaustive search. As you can see, obviously, it goes up exponentially, whereas our running time of our heuristic search remains reasonable. Actually, it's linear, hopefully linear. And the memory expense is the same. As you can see, you can go up to 9 gigabytes if you are not careful enough, if you use the original one. And then in practice, when we really look at the performance of our method, of course, this is the original method when a lot of things are missing. And this is our method, but we have things that are wiggling, right? And this is our final result that gives us a reasonable, better estimation of the mesh. Things are stretched out as we want. And then here are some other numbers, and this is our final mesh. Okay, these are the papillary muscles, and then these are the trabeculae of the final result. And usually, we want to estimate this trabecula when the heart is fully relaxed, because only then the trabeculations are fully stretched. If you imagine when the heart contracts, those things are touching each other. There's no way we can figure them out accurately, right? So that will beg for another question that I will explain a little bit later. And the image size usually is 500 by 500 by 300. But really, when you localize to the ventricle, it's just up to 100 cubic. And then here's a spatial resolution. And then we select the threshold to be 80, and then the total running time, including the persistent computation, is 7 minutes roughly over six different data. And also we have the memory consumption 6 to 10 gigabytes. All right, and yes, gigabyte.
Oh, no, GT.
Oh, ground truth. Sorry, yes. So basically, we take the mesh. We also take the ground truth mesh and see how far is our mesh from this ground truth mesh. Of course, the bigger the number is, meaning the more error, the more mistake we're making. So if we use the regular segmentation methods, here's the average error in terms of pixels, voxels, sorry. And then this is a naive one without the optimization, and this is without a localization solution, and this is our result. Okay.
Is that over the entire heart, or just the little spindly bits, the trabec?
No, over the entire heart. Yes. But this standard deviation is over different data, 6 different data. I mean, we have more, but yeah. We also carried out some small morphology analysis. So we take the whole heart, right, and we look at different parts of the whole ventricle, different parts of it. And you can actually observe, when you get near to the valve, the trabeculation look more like a vertical and thicker. Okay? Whereas near the bottom, which this is called apex, the trabeculation is more like a net meshes. And they're fine, and they're like not in parallel. And these are a couple of big muscles. These are called papillary muscles. They're responsible to make sure the valve doesn't flip over with certain strings attached, basically. Right? So, and these are consistent with the real heart observation. You can see near the valve, you have the strong muscle columns and roughly parallel. And then near the bottom, you have very fine ones. And this is, and certain area you would have almost trabeculation free when it's near the separatum between the two ventricles. And finally, there are a few further questions. First of all, as I said earlier, when the heart is fully relaxed, we have reasonable confidence that we can estimate how the trabeculation look like. But when the heart contracts, we have no idea. So then what we need to do, the solution is, we start with a mesh of the relaxed heart. We track the mesh through the time sequence. Okay, actually, what we have is, for each CT image, is actually 10 frames over the whole heartbeat. Right? So we track those mesh through these images. During the process, we try to preserve the topology of the original mesh. That's one of the projects we're working on. Okay, the hope is eventually we will have a full 4D model of the heartbeat that give us flow estimation, all kind of things we can do. And also, as we said earlier, the way we validate it so far is really we just make a student staring at the screen forever and then try to figure out whether this thing is real or that thing is real. Right? We claim they are domain experts, but really domain experts wouldn't have the time, so we have students actually do it. Right? So what's really happening is we have this particular idea in the sense that we take a specimen, dead human heart, and then scan them. The way we scan them is we can actually scan them with a really long time over the night using MRI scan. Then we can get really high resolution so that we can take the threshold. Then we already have the truth, ground truth, reasonable ground truth. And with that, we can throw in all kinds of simulation, the noise, the different image modality, and then try to see how our method is working with those different settings. And hopefully that gives us a good estimation of at which stage, at which parameter setting our method is more reliable. Of course, there are limitations. What are the limitations? First of all, when the heart is dead, what's going to happen is the things are not fully stretched. The trabeculae will, many places, the trabeculae will shrink. Right? So that also causes an additional problem. So we have to only find the places where we think they are reasonably reliable, and then work on those parts, rather than the whole heart. Okay, and we are currently also scanning some other hearts with certain different pathologies, and see under different conditions how are those trabeculae different. Okay, hopefully this evidence will help us in practice. Okay, so just to summarize, we have cardiac image segmentation, and then the basic idea is to apply this topology prior to help us figure out this trabeculae, and then use them for us to reconstruct it in a high accuracy. And then the additional problem is sometimes we need to find the optimal cycle representing these handles. And then we use, in practice, the original algorithm doesn't quite work, so we have to design certain A-star search algorithm to make it efficient. Right? The question is, can we extend this method to a more general setting to other applications? The answer is yes. So here are different works we have done. Very quick, brief review. So in real image, you have a lot of cases where you have very fine structures. In this retinal, you have these vessels, and this is a neuron. You want to figure out those very fine structures. And using topological methods can help you in this context. If you think about it, the philosophy is very similar, where the origin method will always try to cut it off, because the way once you cut it off, it's just always cheaper to cut it off. Right? So the question is, our method, can it try to figure it out first and force it to be part of the solution? Right? And here's another application. We designed this stencil art technique where you have the original image. The final goal is design a set of stencils. Each stencil is one either cupboard or some metal, then you cut some holes out of it. And then you put it on the wall, you spray paint different colored paint through it. The hope is when you apply them one layer by layer, the final result is some nice approximation of the original image. Okay, our idea is to help people design these stencils for the given input image. All right, you can design the stencils, formulate the stencils as something similar to the image segmentation tasks, but the actual constraint is the following: each stencil, after you cutting the hole through, the remaining part has to be connected. Imagine if this piece is disconnected to the rest, when you move things around, it's really hard to make sure it is in the right position. That's a problem. Okay, and so because of that, we have to enforce this topological constraint, connectedness constraint in this framework. In particular, you can see to make sure this nose piece connects to the rest, we are actually sacrificing the performance of the final result. That's the price you have to pay. The question is, where should I make this bridge? Right? And here is the way where we can pay the least cost. Right? Of course, otherwise I can totally make a bridge horizontally, then it's more unnatural, the final result look like. Right? So our algorithm is trying to figure out where is the right place to connect things. And the philosophy is the same as previous. And here's the real examples where we design the stencils, we cut them out using a laser cut machine, printer, and then we apply them in real life. Finally, I want to make a note about the homology localization problem. It's not really just the trabeculae. I had this discussion with Michael over the dinner, and tried very hard to convince him. I still don't know whether it was successful or not. So here's one example application we have done over the past few years. This is the neuron image segmentation problem. Okay, the input are electron microscopic image. Each pixel is nanometers, like 5 or 6 nanometers squared size. And this is from the different small animals' brain, either fruit fly or a mouse. Okay, and then this is what you can see. The goal is to cut this into different regions so that each of them correspond to a neuron cell.
This is a slice.
Yes, this is a slice. But actually what you get is a stack of slices. And then the goal is you try to connect all those different slices. So final result are a 3D stack, a 3D set of all different cells. You can understand how these neurons are interacting with each other. Okay. But the first step, it has to get this thing cut off, right? So we have very advanced technology. In particular, you can use deep learning, convolutional neural network, to learn a very good likelihood map. This likelihood map, the darker pixels, meaning it's more likely to be the membrane of the neurons, right? Separating different regions. So you can see a very nice result already, right? If it's clear boundary, you know how to do it, right? Over here, you know this is the boundary, and these are small cells as well. But the problem always comes with this blurred region. And over here, you can see also the blurred region. This is where you don't know. You're not so sure. Should I cut this into, should I make a separation over here or not? And persistence actually comes handy in this context. Okay, this is final slides. Just one quick moment. In this case, if we want to make the right separation, we have to develop features. And these features really not only depend on persistence diagram itself, we also need to actually go back, extract the corresponding handles of these persistent dots, and extract geometric and appearance features, and use them to help us determine whether this thing is a real thing or not. Again, the basic philosophy is homology, topology, persistence can help us generate reasonable hypotheses. And then the next step is use those hypotheses to extract different discriminative features and help us in the next step. That's my general pitch in this different context of machine learning. With that, I'll conclude. Thank you. Yes?
Can you explain how you force your binary segmentation algorithm to take into account these topological cycles to include them?
Yes. That's actually a good question. So the point is, in the data term, you pay at every pixel. Once we figured out, okay, this loop has to be included, I just force it to be very expensive if you do not include it in the final result. That make sense?
Yeah. Would that not give you something like sort of likely to be infinitely thin though?
Along this cycle, each voxel, if you classify it into the background, you will pay very high price. And then the model will basically, the algorithm will give me the right answer. Yes?
So the mesh on which you apply the algorithm, is it a surface mesh or a superficial complex?
We apply this on the cubic complex of the original image rather than the mesh. It's not the mesh.
So it's a 3D triangle.
3D cubical complex. It's supposed to be triangulation, but that's more expensive. So we actually use a cubical complex, meaning each voxel is a node, and then we take edges, horizontal edges and vertical edges, and the square faces and the cubes.
But why did you try it on the boundary, on the surface?
You have to start with the surface in that case, right? We don't have the surface yet when we try to figure out where the homology is.
But if you have the 3D cubical complex, you can always take a boundary, which is a surface. It depends whether it is a complex or not.
That could be a reasonable question. For example, I can take a different threshold and then focus on one particular homology and localize the shortest cycle within that mesh of it. That could be possible to try out.
Because then the complexity will go down dramatically.
Yes, that makes sense. Yes, that makes sense. I'll be interested to try out. Yeah, thank you.
Do you have software available for finding the shortest?
Yes, we are planning to publish it within a few weeks. It's supposed to for general context as well, including RIPS complex. But of course, the computational persistence is kind of less efficient, and we're relying on other people like Uli and Michael to do it. Yes, it makes a lot of sense. I just didn't get my hands on those data. Yes.
Would the method for optimizing the rock-centered cycles generalize to higher dimensions, higher homological degree?
No, only one-dimensional cycle.
With finding the longest cycle, one thing that I would be worried about is that.
You mean the shortest cycle? I'm sorry.
The shortest cycle. Maybe that at the birth time, you don't get. If you go slightly past the birth time, you might have a much shorter cycle.
Yes, we do have a design, a theoretical question along that line. Can you find the shortest cycle with a little bit offset of the homology? That would also make sense. And another thing that could make sense is, can you find some kind of media axis? Because do I have it? It's just, yes, maybe the shortest one is not necessarily the right one. Maybe I want something in the middle of the current status of the trabeculae and things like that. All of them are very legal questions. And the question is also relevant in terms of tracking. Because when you track, you will also look at how the function deforms. And then you need to consider certain kind of offset of the functions and the level sets. It's also relevant. Yes?
When you were choosing some thresholding for this persistent diagrams.
Yes.
If you use pictures from different machines, which is calibrated a different way, do you have to go through this whole process of choosing the thresholding?
We assume our data is from the same setting. We assume that's what you're saying is the right question. If in that if it's what you're saying, then you have to normalize it properly. And then that will be
sometimes it's very difficult to those are not comparable, right?
There was a reason I focused more on medical images, because in those cases, in general, the setting is more fixed. The set of data you get has a same set of parameters. For natural images, and nowadays when people try to gather a lot of different data from online from different resources, that's a bigger issue. But that's a big issue for general image analysis and medical image analysis completely. Yeah, but would be a very interesting question if we have a particular case, and then we can study how to solve it properly and validate it. Any other question? Yes?
If I had multiple scans of a single color, can I somehow refine the mesh?
I don't know. We do have issues in the sense that when a person takes the images, right, not only you have the heart beating cycle, but also you are breathing. Your body is actually moving. And there are a lot of different dimensions you're talking about. We are working on some MRI data with this kind of really high dimensional, different parameter settings. How do you reconstruct the whole actually manifold of the hearts along that line? But I'm not sure on the CT context.